Hi everyone, welcome to week number 5. We have passed the halfway mark of the term now, and so we're on a downhill slope. And let's get going with week number 5. So, a recap of last week, you learned about cellular respiration, and cellular respiration is producing ATP, the energy currency of cells, the most important energy molecule. So it's the production of ATP by the breakdown of food by the use of oxygen. Our experiments went crazy. Those germinated peas did a tremendous amount of cellular respiration very quickly. I think they could have shortened that experiment to 20 minutes instead of 10. 10 minutes instead of 20, sorry. Because by the time I reached the 10-minute mark, my dye had run all the way up past the 0 to the -1. So I think a lot of you got the same results that I did. So we studied cellular respiration and the production of ATP. It's tremendously important for us because this is the fuel that runs all the reactions in our cells. The topic this week is DNA, one of my very favorite subjects. DNA, even have this, been sitting back here on my printer every time I've talked to you. Love me some DNA, deoxyribonucleic acid. It's the genetic information in cells. So the instructions to make an organism are found in the DNA, in every organism. Reminder, viruses are not organisms. Viruses do contain genetic information in the form of either DNA or RNA, never both. So that old coronavirus that everybody's talking about now, the reason that we're all stuck inside is an RNA virus. But this week we're talking about DNA. It's the genetic information in all organisms, whether you're a bacterial cell or you're as big as us or you're a plant or mushroom or a little amoeba crawling around in lake water. DNA determines what you are. The code for the production of all the necessary proteins and enzymes that your cells need are found in the DNA. So it's a terribly important molecule. And of course, we know a lot about DNA these days. I know that you do, even from just what you hear on TV. So, for example, in forensic science, a hair was found at the scene of a crime. Oh, let's do DNA typing on it and see if we can figure out who left that hair behind. So it's just in the news all the time. What is your genetic predisposition for certain diseases? Or what's your genealogy? Send in your DNA and we can figure out who your ancestors were. So there's just a lot in the news all the time about DNA. It's a terribly important subject. It's going to be even more important for you in the future because your medical care will be based on your DNA, largely. You will have a chip on a card that you give to your doctor, which will have your genetic code, and that will determine some of the treatments that you get because of the genes that you have present in all of your cells. Okay, let's talk about the lab this week. The lab this week is a very quick lab. It tells you in the lab that it's going to take you about an hour. It didn't take me an hour. It was very fast. Cheek cell DNA extraction. Every time you see forensic science on TV, you see people swabbing the inside of somebody else's mouth. That's called a buccal swab. Buccal, like that's on your belt. A buccal swab, but it's not. It's B-U-C-C-A-L, buccal. It means your cheek. So they swab the inside of your cheek to get cheek cells, just like we're doing in this lab, and they isolate the DNA and then look at the characteristics of it. So this is a good lab for you. It shows you that you really can get a lot of DNA out of a few cheek cells. One thing that I want to mention to you is that it tells you to measure out 2.5 milliliters of the sports drink. Well, your cup is measured in cc's. cc stands for cubic centimeter, And I don't know if you remember this, but 1 cubic centimeter is equal to 1 milliliter. Now these are the tiniest markings on that cup. 2.5 milliliters is a tiny amount of the sports drink, so just realize that. You'll have to look for that marking very closely, is right at the bottom. Another thing that you need to be aware of is that your ethanol, in order for this to work, has to be extremely cold. Now they say put it in ice. I don't think that that is cold enough. What you should do is take that little tiny bottle of ethanol and put it in. It is denatured ethanol, by the way. Don't drink it. It will make you sick. Just as a side note, put your ethanol in your freezer overnight, not in your refrigerator. Put it in your freezer. The colder it is, the better this experiment works. Okay, so leave it in your freezer until right at the point when you need it, and then bring it out, and it will help to precipitate your DNA out of your cheek cell lysate solution. Lysate just means that you broke open the cheek cells to release the DNA. That's the way that you're going to get to the DNA. And like I put on here, it's a fairly quick lab, so that's good for you this week. Yay! Because some of our labs have been kind of long. You'll also do the PCR simulation this week. It's a good one. I like this one. And you're going to answer the questions about PCR. I'm going to explain a little bit more about that in just 1 second. And you're going to need to turn in milestone number 2, which is your experimental design. So you're going to tell me the whole plan of how you're going to do this experiment that you've told me about in milestone 1. Please go back and read the information that I've written in the rubric about your milestone 1. You may have to make some adjustments to your experiment based on what I have read. So please go back and read that first before you design this experiment. Otherwise, the experiment may not be any good. So do that. And if you have any questions, like how in the world am I going to figure out how many pockets of salt water I have in this ice, Dr. Sigmon? What am I going to do? I've got some ideas for you. We can come up with something. There's always something that you can do. And it will be something simple that you can actually do, I promise you. So please do ask me questions if you have any on this experimental design. One thing that I want to mention, because I've seen a lot of this, I want you to put this in the correct format. It tells you to put this in outline form or in bulleted points. So you're not writing this out in paragraphs. You weren't supposed to do that with milestone 1 either, but I didn't take off any points for that. But now you should be putting this in the correct format. I don't think you're going to be using any references for this because you're really telling me in step-by-step fashion how you're going to do this experiment. Okay, so just an outline, step by step. That would be perfect for this. That's milestone 2. Okay, then I did want to talk about PCR for just a minute. PCR is the polymerase chain reaction, and if you noticed in the tip that I just put online, I think it was yesterday, the testing that they're doing for the coronavirus all across the world is based on this technique, PCR, the polymerase chain reaction. And so we're looking to see if viral RNA is present in swabs from people who might have been infected through the use of the PCR technique, polymerase chain reaction. This term just means polymerase is the enzyme that makes a polymer. The polymer that we're interested in is DNA. And you can see chain reaction, right? It happens one step after another, and it gets bigger and bigger and bigger and bigger. That's all this term means. There are 3 basic steps to this process: denature, anneal, and amplify. So let's talk about what they mean for just a second. Denature. So this is at a high temperature. you denature the DNA that is present in your sample. So what you're doing is you're breaking apart the two strands of the DNA. You're breaking this ladder apart right between those nucleotides. That's the denaturation stage. The second stage is annealing. So you have to add primers to get the copying process started. Primers are just little pieces of either RNA or DNA that start this process. So attach the primers to either side of the DNA right where you want it amplified or copied. Okay? So this is just to get the process started. So annealing is going to happen at a lower temperature. Denaturation takes place at a high temperature because you got to break bonds. Annealing, you want to make bonds again. So you're going to cool down your DNA solution again, and then amplify. That temperature is based on the temperature that's preferential for the polymerase enzyme. Polymerase is an enzyme that makes the new DNA. How does it do that? It attaches nucleotides, the new little subunits of DNA, to those primers in a step-by-step fashion. So the 3 steps: denature, break apart the DNA; anneal, add the primers to the DNA; amplify, make the new pieces of DNA. So this whole process is just taking very, very, very tiny quantities of DNA and making lots and lots of copies of particular parts of the DNA that you want to look at. That's the reason that you do PCR. So I want you to understand the basics of it before you get bogged down in that simulation and you're really not understanding what in the world is going on. It's a tremendously important technique. You can pick up any biology journal right now and you will find at least one article in any biology journal that you look at that has used PCR. It is just out there. It is so important, and it is so important for diagnosing something like a viral infection, because viruses take over your cells. They just don't constantly float around in your bloodstream, because they attach to your cells and get inside. So the only way that you're going to find out if viruses are present is to look for that genetic information. And we are seeing this in action right now as all of us are sheltered in our homes. We see this particular type of testing being done all around the world. So that is PCR. And then next week. So next week's lab isn't going to take a tremendous amount of time, but it is either going to take 7 or 8 days to grow your plants from seeds, because you grow in 2 sets of seeds. Okay, so the sooner you can get this started, the better. Because like I said, the shortest amount of time this is going to take is 7 days. So if you start on Sunday, you're going to be finished on Saturday night, and then you got to write up that lab report. So if it were me, I would do the labs for this week right now and get those done. The simulation is nothing much. You just have to answer questions after you watch the simulation. And like I said, the cheek cell extraction doesn't take long. So I would start the seeds as soon as possible so that you have plenty of time to write up your lab for that week. It's going to take a long time for these plants to grow, even though they grow really, really fast. I'm not saying that it takes a long time for them to grow, but you're growing 2 different sets of seeds. So it's going to take a long time for you to get through the whole growth process of this experiment. So I'm just warning you about that ahead of time. If you want to start at the end of this week, that would be awesome. And then you have plenty of time for your lab report. If you have any questions, if you have any issues, if you're having any trouble getting your work done and you need an extension or something, please let me know. I'm here for you. Email me at m.sigmon@snhu.edu. I'm here for you. I'll be glad to help you. And I hope you have a wonderful week. Even though you are sheltering at home, at least you have time to get your work done. So I hope you have a really good week. And, oh, I forgot to tell you, I changed the two pictures in the background. There's a monarch butterfly with the zinnia, and then one above it. I'm going up, 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 up. That's a whale tail. I took both of those pictures, of course, and that's a whale tail from some of my adventures last year. My sister just sent me a message yesterday about our trip to New England, out from Boston, Gloucester, Massachusetts, when we went to watch the whales 3 different times last year. And it was about this time last year when we went for the first time, and here we are all stuck at home right now. So we reminisced a little bit about our whale watching. So anyway, I hope you have a great week, and let me know if you need anything.